All right, get ready to dive into the world of trypanosomes. These microscopic parasites cause some serious diseases, like sleeping sickness and Chagas disease. And they have some pretty unusual biology. We're talking cutting-edge research that could change global health, so buckle up.
Sounds exciting.
It is. Okay, so trypanosomes, they're single-celled parasites. They impact millions around the world, mostly in the Southern Hemisphere. And, with the climate changing, their reach could expand. So this research is super important.
Definitely. A lot at stake.
We'll be looking at some groundbreaking work from EMBL Grenoble, where scientists have mapped out this protein complex. It's vital for trypanosomes to survive. This could be our ticket to new drugs.
Interesting. What kind of protein complex are we talking about?
Well, first, a quick recap on how genetic info works. DNA turns into mRNA. Then mRNA gets translated into proteins. Trypanosomes, they're eukaryotes like us, so they follow the same basic plan. But their mRNA processing is different, which gives us a unique target for drugs.
Right. You target the differences, and you can knock out the parasite without harming human cells.
Exactly. So tell me more about this target, this protein complex. What's the deal with it?
It's called the nuclear cap binding complex. It's like a landing pad on the mRNA, a platform for other factors to interact. It guides the RNA through processing. Without it, the mRNA can't function, and the parasite dies.
So it's like a quality control checkpoint for the mRNA.
You could say that. But here's where it gets interesting. Humans, we have 2 subunits in this complex. Trypanosomes have 4. And the function of 3 of them has been a mystery, making this complex, well, a bit of a puzzle.
Double the subunits, double the trouble, I guess. But if it's that important, wouldn't that make it really hard to study? How did they even figure out the structure?
Well, it was tough. It's very flexible, this complex. It has this 2-lobed structure, one part similar to humans, but the other lobe, it's connected by a really flexible protein. Imagine trying to study a jellyfish.
A jellyfish?
Yeah, so they used cryo-electron microscopy, which gave them a snapshot, but the flexible parts were blurry. To fill in the gaps, they used small-angle X-ray scattering. Basically, they shone a powerful X-ray beam at the complex and analyzed how the rays scattered to figure out the shape.
Clever. 2 methods for a complete picture. Impressive. But what did the structure reveal? What did they learn about how it works?
They found a second RNA binding site. You know how mRNA has a cap at one end? Well, in trypanosomes, this cap is heavily modified. The complex binds to this, but they also found another binding site for RNA. Totally unexpected.
A second binding site. So there's more to it than just the modified cap. This is getting interesting. What does the second site bind to?
This is where things get even cooler. It's on one of those mysterious flexible subunits, and it binds specifically to double-stranded RNA.
Double-stranded RNA. Isn't that usually assigned to something weird like a virus?
You got it. We don't know exactly what it means, but the fact that it binds to double-stranded RNA suggests it might be involved in detecting threats or regulating gene expression in a way we haven't seen before.
So maybe it's like the trypanosome security system, or even its control panel for genes.
Possibly. And don't forget, this whole complex is significantly different from the human version, making it a prime drug target. Imagine a drug that messes with this complex in trypanosomes, but leaves human cells alone.
A magic bullet for these diseases. And I heard there's a PhD student diving into this right now. That's fantastic. But it seems like this research is bigger than just drugs.
Yeah.
It impacts more than just finding a cure.
Absolutely. Trypanosomes, they're not just parasites. They can help us study eukaryotic evolution, the history of cells with a nucleus like ours. They've been around for a long time. By studying how their cells work, we get clues about how all eukaryotic life, including us, came to be.
Wow. So these little parasites could unlock secrets about all life on Earth. Pretty amazing. Hold on a sec. You mentioned the lead scientist is also getting funding to study something called transplicing. What is that? How does it fit into all of this?
Transplicing. It's another unique mechanism in trypanosomes. Basically, they can take pieces of RNA and stitch them together, like tiny genetic engineers. And here's the kicker. Scientists are thinking about using this transplicing machinery in humans for therapy.
Wait, are we talking about using what we learned from these parasites to develop gene therapies for humans?
Exactly. It's early days, but imagine correcting a faulty gene by introducing a new piece of RNA that gets transpliced into the existing mRNA. fixes the genetic defect, a whole new way to approach gene therapy.
That sounds like something out of a science fiction movie. But we were also talking about climate change. How does that play into all of this?
Right. As the planet warms, these insects that carry trypanosomes, their range is going to expand. So diseases like sleeping sickness and Chagas disease, they could start popping up in new places, affecting more people. We need to be ready for that.
So this research isn't just about scientific curiosity. It's about being prepared for a future where these diseases might become even more widespread. It's about protecting global health in a changing world. And it all starts with understanding these fascinating and potentially dangerous little parasites. But let's get back to that double-stranded RNA binding site for a moment. What are some of the theories about its function?
One idea is that it's a viral detector. When a virus infects a cell, it often makes double-stranded RNA. So this subunit could be like a sensor triggering a defense mechanism in the trypanosome.
Like an internal antivirus program. Clever.
Exactly. Another theory is that it's involved in gene regulation. Double-stranded RNA can also form naturally from the cell's own RNA, and that can be a signal to turn genes on or off.
So this one subunit could be controlling which genes are active in the trypanosome. Multitasking at its finest.
Right. Gene regulation is crucial for so many processes. This finding opens a whole new door for research. How do trypanosomes control their genes?
Incredible. We started with a potential drug target, and now we're talking about viral sensors and gene regulators, all in this one complex.
And that's just scratching the surface. These parasites are complex and fascinating. We're just starting to understand how they work.
My head is spinning. We've covered a lot. The nuclear cap binding complex, new drugs, gene therapies, evolution, even climate change.
It's a lot to take in, for
Right.
sure.
But hold on, there's more. In part 2, we'll go deeper into transplicing for gene therapy. We'll look at the challenges and the ethics of using this parasitic power for human health. Don't go anywhere.
You know, this research on the nuclear cap binding complex, it's really fascinating. It makes you think about bigger questions, like how did eukaryotic cells, cells with a nucleus like ours, how did they evolve their complex machinery?
That's a great point. It's easy to focus on the immediate implications, like new drugs. But you're right, this research could change how we understand life itself. So how do trypanosomes fit into that?
Well, trypanosomes, they're ancient, right? Millions of years old. They represent an early branch on the eukaryotic tree of life. By studying their cellular machinery, we can get clues about those first eukaryotic cells, what they might have been like.
So, like a living fossil, giving us a window into the past. But how do we know their machinery hasn't changed a lot over millions of years?
Good question. Of course, they've evolved. But a lot of their core cellular processes are surprisingly similar to those in more complex eukaryotes, like us humans. That's why they're so valuable for studying evolution. They give us a baseline, a starting point for understanding how more complex systems developed over time.
So by comparing their machinery to ours, we can trace back the evolutionary steps that led to all the diversity we see today.
Exactly. And what's really interesting is that while some things about trypanosomes are unique, like they're transplicing, other things are very similar to us, especially the really basic processes like DNA replication and making proteins.
Like a mix of old and new features, a glimpse into both the past and present of evolution. Amazing. But I have to admit, this transplicing thing, that's what really caught my attention. It sounds like science fiction. Can you remind me what it is and why it's so special?
Imagine a cell that can cut and paste RNA together, creating new mRNA molecules that actually work. That's transplicing. They have their own RNA editing system built right in.
So instead of just translating the mRNA directly from the DNA, they're shuffling things around. What's the point of that? Why do they do it that way?
That's a mystery scientists are still trying to solve. One theory is that it lets them make a lot of different proteins from a smaller genome. Another idea is that it helps them regulate gene expression in a more complex way.
So it's like a genetic toolkit, giving them more flexibility and adaptability.
That's a great way to put it. And the really exciting part is that scientists are looking at whether we can use this transplicing machinery in humans for therapies.
Now we're back to gene therapy. Can you walk me through how that could work?
Okay, imagine you have a gene with a mutation that's causing a disease. With transplicing, we might be able to introduce a new piece of RNA that gets spliced into the existing mRNA, correcting the problem, editing the mRNA directly instead of replacing the whole gene.
So it's like a molecular patch, fixing the code at the RNA level. That would be a lot less invasive than traditional gene therapy.
Exactly, and that's why everyone's so excited about transplicing. It could be a safer, more precise way to treat genetic diseases.
But there must be challenges, right? It sounds almost too good to be true.
Oh, there are always challenges when you're trying to turn basic science into actual therapies. One big hurdle is figuring out how to deliver this transplicing machinery to the right cells in the body. And we need to make sure it's safe, that there are no unexpected side effects.
So still a lot of work to do before this is ready for patients. What are the next steps in this research?
Well, the lead scientist got funding to dig deeper into transplicing. How does it work in trypanosomes? Can we adapt it for human cells? It's a long road, but the potential is huge.
It's incredible to think that studying these tiny parasites could lead to revolutionary treatments for genetic diseases. But let's circle back to climate change. We talked about how rising temperatures could spread trypanosomal diseases. What can we do to prepare for this?
Well, research is critical. We need to know these parasites inside out, develop new drugs, new diagnostics, and ways to control those insects that transmit the parasites.
A multi-pronged approach, attack the problem from all angles. But research takes time, and climate change is happening now. What can we do in the meantime?
Public health education. Big time. Especially in areas where these diseases might spread. People need to know the risks and how to protect themselves. Using insect repellent, wearing the right clothing, getting medical help quickly if they have symptoms. Simple things can make a difference.
So education is key, empowering people to take preventive measures. But can we do anything on a larger scale?
Absolutely. We need to tackle the root causes of climate change. Reduce those greenhouse gas emissions, switch to renewable energy, protect biodiversity. These are all crucial steps to mitigate the effects of climate change, including the spread of these diseases.
So it's bigger than just fighting parasites. It's about tackling the environmental problems that are making things worse, a real intersection of science and policy. It highlights how everything's connected, our health, the environment, the choices we make. This deep dive has been packed with information. But there's one more thing about trypanosomes that I find fascinating. You mentioned they have a unique way of responding to threats. What kind of defense mechanisms do they have?
Well, they have this amazing ability to change their surface coat. Trypanosomes are covered in this thick layer of proteins. It's called the variant surface glycoprotein, or VSG.
A coat of armor. How does that help them?
This VSG coat is incredibly diverse. Thousands of different VSG genes, and they can switch between them. They change their coat like a chameleon changes color.
They can make themselves invisible to the immune system.
That's it. When the immune system starts to recognize one VSG coat, the trypanosome just switches to a different one, disappears again.
A constant game of hide and seek with the immune system. Pretty clever. But how do they switch their coat like that?
They have this sophisticated system for regulating their genes. They only express one VSG gene at a time. And they can switch between these genes randomly. The immune system can't keep up.
So they're not just evading the immune system, they're outsmarting it. But there must be a limit to how many times they can do this, right?
That's the million-dollar question. Scientists are still trying to figure that out. We know they can switch thousands of times, but whether there's an upper limit, we don't know yet. And that's one reason why these diseases are so hard to treat.
Masters of disguise and incredibly resilient. These parasites are tough opponents. This is why research like this is so important. We need to understand them to beat them. Oh, wow, look at the time. We're almost out of time. And we still need to discuss the ethics of using transplicing for gene therapy. We can't leave that unexplored. Join us for part 3, where we'll delve into the potential benefits and risks of this revolutionary technology. Okay, we're back, and we're still buzzing about trypanosomes. Remember, transplicing, that wild ability to stitch RNA together. Scientists are seriously considering using that for gene therapy in humans.
Yeah, it's super promising. But like with any powerful new tech, there are questions. Big ones.
Right, ethical questions. So let's talk about those. What are some of the ethical concerns with using transplicing for gene therapy?
Well, safety, for one. I mean, we're messing with the very foundation of life here. Before even thinking about using this in people, we need to be absolutely certain it's safe. No unintended consequences.
Makes sense. We've seen issues with other gene therapies, unexpected side effects. So safety first, for sure. What other ethical considerations are there?
Accessibility. Gene therapies can be expensive, complicated. We need to make sure any transplicing therapies are available to everyone who needs them, not just a privileged few.
It's not just about the science itself, it's about fairness. Everyone deserves access to these potentially life-saving treatments. Are there other ethical dilemmas we need to think about?
Some people are worried about misuse. What if this tech is used for genetic enhancement, creating designer babies with specific traits? That's a line we need to be very careful not to cross.
Wow, that's deep. It makes you question what it means to be human. This goes way beyond science. We need ethicists, policymakers, everyone involved in this discussion. The ethical side of transplicing is complex, for sure, but let's not forget the potential good. We could be talking about cures for genetic diseases that have been around forever. Isn't that a goal worth pursuing?
Of course. The possibility of ending suffering, it's huge. But we have to be careful, responsible, make sure this technology is used ethically.
Balance is key. Weighing the potential benefits against the risks. We need to talk about this openly and honestly, and we need to do it now, before this technology becomes a reality. This deep dive into the world of trypanosomes has been incredible. We've learned about their strange biology, how they can help us understand evolution, and the potential threat they pose in a changing world.
It really shows how powerful scientific curiosity is, and why we need to support fundamental research. You never know what you might discover.
Exactly. Who knew that studying these tiny parasites could lead to new drugs, gene therapies, and even a deeper understanding of life itself?
It proves that every living thing, no matter how small, has something to teach us.
Couldn't have said it better myself. That's a great place to wrap things up. Thanks for joining us on this deep dive into the world of trypanosomes. We hope you found it as fascinating as we did. And who knows, maybe this has sparked your own scientific curiosity.
Maybe there's a future scientist listening right now.
That's what we like to hear. So keep learning, keep exploring, and keep asking questions. You never know what amazing discoveries are waiting out there.
Yeah.